Protein denaturation is a process that alters a protein's natural three-dimensional structure, leading to a loss of its biological function. While the primary sequence of amino acids, which are the protein's basic building blocks, remains intact, the higher-order structures, secondary, tertiary, and quaternary, are disrupted. Proteins maintain their precise shapes through various weak interactions and bonds, including hydrogen bonds, which form between polar side chains and within the protein backbone. Hydrophobic interactions occur when non-polar amino acid side chains cluster together in the interior of the protein to avoid water. Ionic bonds, also known as salt bridges, form between positively and negatively charged side chains. Disulfide bonds are covalent bonds formed between sulfur atoms of two cysteine residues, providing strong stabilization, particularly in extracellular proteins. Van der Waals forces are weak interactions that fine-tune protein structure. Denaturation occurs when these fragile interactions are broken or significantly altered. The primary consequence of denaturation is the loss of a protein's specific three-dimensional shape. Since a protein's function is intricately linked to its structure, denaturation typically leads to a loss of biological activity. Enzymes lose their catalytic activity as their active site is deformed. Structural proteins lose their ability to provide support or form stable structures. Transport proteins cannot bind and transport their specific molecules. Denatured proteins often expose their hydrophobic residues, which were typically buried in the protein's core, to the aqueous environment. This can lead to aggregation or precipitation of the denatured proteins, making them insoluble. Denaturation can be reversible. If the denaturing conditions are mild and the primary structure remains intact, some proteins can renature or refold back into their original functional conformation once the denaturing agent is removed. This suggests that the primary amino acid sequence contains all the necessary information for proper folding. Irreversible denaturation happens in more extreme conditions where the denaturation is permanent and the protein cannot regain its native structure and function. This often occurs due to extensive unfolding, aggregation, or covalent modifications that prevent proper refolding. A fried egg, for instance, cannot be unfried because the denaturation of its proteins is irreversible. Various factors can induce protein denaturation. Increased kinetic energy at elevated temperatures causes molecules to vibrate more rapidly, disrupting hydrogen bonds, hydrophobic interactions, and van der Waals forces. This is why cooking an egg turns the clear liquid white and solid. Extreme pH levels alter the charge states of amino acid side chains, disrupting ionic bonds and hydrogen bonds crucial for maintaining protein structure. For example, adding lemon juice to milk causes it to curdle due to protein denaturation. Urea and guanidine hydrochloride. These chemicals interfere with hydrogen bonding and hydrophobic interactions, leading to protein unfolding. They are often used in laboratory settings to study protein folding. Organic compounds like alcohol can disrupt intramolecular hydrogen bonding within the protein. They do this by forming intermolecular hydrogen bonds with protein molecules. Heavy metal ions like mercury, silver, and lead form strong bonds with carboxylate anions of acidic amino acids or sulfhydryl groups of cysteine. This disrupts ionic bonds and disulfide linkages. Detergents and soaps disrupt hydrophobic interactions because they are amphipathic, meaning they have both hydrophobic and hydrophilic parts. Mechanical agitation, such as vigorous shaking or stirring, can physically disrupt the delicate balance of forces holding the protein structure together. Whipping egg whites to create meringue is a common example of mechanical denaturation. Radiation, like UV radiation, supplies kinetic energy to protein molecules, causing them to vibrate rapidly and disrupt. Protein denaturation is a common phenomenon observed all around us. Boiling or frying an egg. The clear albumin protein in egg white denatures and coagulates, turning opaque and solid due to heat. Cooking meat. Heat denatures the proteins in muscle fibers, making the meat firmer and changing its color. Making yogurt or cheese acids produced by bacteria denature milk proteins, causing them to curdle and thicken. Perming and straightening chemical treatments and heat denature and reform the keratin structure of hair, changing its shape. Curdling of milk, adding acidic substances like lemon juice or vinegar to milk causes the proteins to denature and precipitate. Sterilization heat is used to denature and kill proteins in bacteria, making medical instruments and food